welcome everyone to OsteoBites. Please feel free to put any questions in the Q&A in the chat as we get going. My name is Christina Iptoma, and I am mom to OsteoAngel Dylan and the Director of Scientific Programs at MIB Agents. And today on OsteoBites, we are talking with Dr. Jason Yustein from Emory University. Dr. Yustein is one of our 2022 Outsmarting Osteosarcoma grant recipients, and he will discuss his funded work on combination therapies to improve immune-mediated approaches for osteosarcoma. Thank you so much, Dr. Yustein, for joining us on OsteoBites today. We're thrilled to have you. And thank you to Vicki for joining as our panelist today. Vicki is an osteo warrior and MIB Agents Junior Advisory Board member. So a little bit more about our guest today before we get started. Dr. Yustein is a pediatric oncologist and physician scientist with significant training in molecular and cellular biology. And during his time as the director of the Ferris D. Ewing Sarcoma Center at Texas Children's Hospital, he recognized the need for improving our understanding of the molecular pathogenesis of sarcomas and the identification of new therapeutic avenues, especially for patients with therapeutic resistant and or metastatic disease. His lab has tremendous interest and experience in merging innovative murine models and patient-derived resources towards garnering molecular insights into sarcoma initiation, development, resistance, and metastatic progression, and translating these findings towards testing novel therapeutic interventions for these aggressive malignancies. His lab has developed and characterized robust metastatic genetically engineered mouse models for pediatric sarcomas, including osteosarcoma, which recapitulate the human disease initiation and progression. He recently moved to Atlanta to join Emory University as a professor in the Department of Pediatrics, Hematology Oncology Division, and as the director of solid tumor translational research at Children's Healthcare of Atlanta and the Aflac Cancer and Blood Disorders Center. So welcome, Dr. Yustein. And some other announcements from MIB before we jump into Dr. Yustein's presentation. In addition to our OsteoBite series, MIB Agents offers many programs and resources for the osteosarcoma community, including Ambassador Agents, which pairs newly diagnosed patients and families with survivor families, Gamer Agents, which gives osteo warriors and siblings a community to connect and have fun, and Healing Hearts, which offers Greek workshops for bereaved families. And also an osteosarcoma resources handbook authored by families and doctors, which is a great resource for newly diagnosed families and walks them through what to expect. All of these MIB Agents programs and resources are free of charge, thanks to generous donors who make it better. And if you'd like to support these and all MIB Agents programs, we'll put a link in the chat. And here are some other ways you can support MIB Agents and our programs now and in September for Childhood Cancer Awareness Month. We have our virtual Outrunning Osteosarcoma event now through September. You can run, walk, cycle. You can do it live in person or virtually with friends. You could do it in one day, or you can just do it over the course of 2 months. We'll put the registration link in the chat if you're interested in doing that. And we're doing our gold bows again for September. You can make and sell gold bows for your mailbox in your community, or you can just order a bow, and that will raise awareness for pediatric cancer and also show that you care. We'll put some information in the chat about how you can get involved with that. And then our episode today is sponsored by the Osteosarcoma Institute, a nonprofit organization led by osteosarcoma experts from top US cancer centers who together are concentrating on the cure for osteosarcoma. The mission of the OSI is to dramatically increase treatment options and survival rates in osteosarcoma patients through identifying and funding the most promising and breakthrough osteosarcoma clinical trials and science. And in addition to advancing research, OSI also provides a free resource called OSI Connect for osteosarcoma patients. Their osteosarcoma experts can discuss available treatments, possible side effects, and provide helpful advice for getting the most out of your visits with your treating physician. This resource is available in English and Spanish and aims to help patients and families find answers to their questions. So thank you, OSI. And I'm going to hand it over to Vicki to introduce herself.
Hi, everyone. As Christina said before, my name is Vicki Hoy, and I'm a Junior Advisory Board member and osteo warrior. I'm starting my freshman year at Villanova studying finance next week, but no worries because I plan to continue being on OsteoBites while I'm there.
Yay.
Glad I worked in my schedule, yeah. And thank you, Dr. Yustein, for joining us today, and I'll give you the floor.
Great. Well, thank you, Christina, and thank you, Vicky, for that kind introduction. And obviously, we're very honored to be recipient of an MIB award. So, as Christina said, it's the title of my presentation, and we're really excited about this avenue. It's a relatively new field for my lab, but while it's new, it's also been exciting from the standpoint of spurring on new collaborations, interactions with not only researchers and faculty formerly at Baylor, but also even here now at Emory and elsewhere across the country. So we're really excited about this particular work. So of course, in terms of cancer development, the idea is that cancer is a genetic disease in terms of, you have a normal cell, and unfortunately the cell undergoes some sort of aberration molecularly, genetically, and then leads to transformation and development of a tumor. But of course, that cancer initiation, while it's a key process, it's not the only thing that's obviously going on in the body, because cancer is within a patient. And one of the things that happens with regards to cancer initiation and development is with regards to immunoediting. And that's kind of referring to the process by which the cancer is eventually kind of escaping the immune system. And it's thought these triple E effects, where initially the host immune system is able to potentially control or regulate or eliminate the cancer. And at some point, there might be an equilibrium between the tumor cells and the immune system. And eventually, over time, the cancer cells kind of learn or develop, or whatever, however you want to phrase it, to escape the regulation or control of the immune system. So this immunosurveillance is obviously a key aspect of tumor development and progression. So as I alluded to before, you have this immune escape aspect, and part of that mechanism, or potentially one of the mechanisms by which the tumor cells escape, is through checkpoint inhibitors, or these things called immune checkpoint inhibitors. And that's kind of shown here on the right-hand side. I think everyone's probably heard about these checkpoint inhibitors, and there's a number of different molecules that are potentially involved with these checkpoints. So again, it's a mechanism by which the tumor cell can kind of resist, for example, T cell killing. And so understanding these mechanisms obviously are key, and it's been a very intense topic for the past decade or more in terms of within cancer biology. So the premise is that can we, with regards to immunotherapeutic approaches for any tumor, but for pediatric sarcomas, can we potentially use the patient's immune system to help fight their cancer? That's become, in this area, as of research has taken off over the past decade, as I've mentioned. So the use of drugs known as checkpoint inhibitors have been integrated into treatments for many cancers, and that's kind of the premise is kind of shown here on the left-hand side in this diagram, where you have the tumor cell and those checkpoint inhibitors that are inhibiting some of the immune cells that can fight the cancer. But then if you introduce these checkpoint inhibitors, such as PD-L1 or anti-PD-1, you allow those T cells or immune cells to become activated and potentially lead to the elimination of the tumor cells. Unfortunately for sarcomas, including osteosarcoma, this really hasn't been proven successful, at least as monotherapy and even some combination therapies. And that's shown here in this table over the past several years, where these checkpoint inhibitors have not really had a significant or great response for a number of different sarcomas, including bone sarcomas such as osteosarcoma. So the question is, of course, how can we potentially improve the outcomes with this type of approach or this type of immunotherapy? So what we thought is that, are there rational combination targeted therapies, and I'll get to that soon, that can alter not only the potentially tumor intrinsic properties, but also the microenvironment that will allow these immunotherapeutic approaches to become more successful for osteosarcoma. So our proposal, and again, very fortunate to have MIB helping us fund these types of studies, is that we're going to use some of our models, which I'll go over soon, these mirroring models of osteosarcoma that Christina already described to some degree, with some of the single agent, but also combinations with checkpoint inhibition, and seeing how potentially efficacious these approaches can be in targeting primary and eventually metastatic osteosarcoma. And then, of course, we can use our models to help hopefully get some insights into how these therapies are altering not only the tumor cells, but the microenvironment. And can we learn how these therapies are modulating, some of the environment that may be contributing to whether it be the sensitivity to the therapy or resistance to the therapy. And then basing those insights, can we then develop even better therapies or identify additional vulnerabilities for targeting osteosarcoma?
And Dr. Yustein, can I just
Yeah.
and ask you just for like a brief kind of background? So from your slide, talking about the checkpoint inhibitors and how they haven't been very successful in sarcoma.
Right.
Can you just kind of talk a little bit about how that, knowing that, how that has informed your approach, this other approach that you're taking? Like,
yeah, so
yeah.
Right. So I think one of the advantages, hopefully with our models, we can potentially get some better insights into why the checkpoint inhibitors alone are not really working. But then I think that's also led us to identify potentially other mechanistic signaling aspects that we can potentially target. And I'll go over a couple of the rationale for a couple of the agents that we've selected, but we're also looking at other molecules, but the rationale behind why we're looking at these particular agents to hopefully improve the ability of the checkpoint inhibitors to work on osteosarcoma.
Great, thanks.
All right, and so of course, we need to have appropriate models. I'm a big modeling person, as Christina already said. Both we try to integrate mirroring as well as relative human models within our research, and this one specifically is using a lot of our mirroring models that we've developed. So this slide is just showing that some of the aspects of obviously there's cell lines that people use, established cell lines, human cell lines, and mirroring cell lines, but patient-derived xenografts, which come from our osteosarcoma patients. But one of the things that we've developed over the past several years is genetic engineered mouse models and resources we've derived from these genetic engineered mouse models. And that's going to be kind of the focus of this seminar for now, and how we're applying these models towards understanding not only the effects of checkpoint inhibitors, but the combination therapies that we're proposing. So first off, I just want to give a little background on genetic engineered mouse models. Basically, what they are is that you can genetically alter mice, whether it be germline so that every cell is altered, or I'll talk about soon more conditional models where you're only altering certain subtypes of cells. So without going into too much detail, but one of the key papers that came out about 30 years ago now was actually a close collaborator of mine when I was at Baylor, Larry Donauer. So he was back in the early 90s. Larry published this paper where he did germline alterations of P53. And so what that means is that he either knocked out both copies of this gene P53, because as you know, each cell has two copies of a gene, one from mom and one from dad. So he knocked out both copies of P53, and that's shown here. And what you can see is that when he did that in every single cell in the mouse, these mice died very early, or very quickly in age, and they usually died from T-cell lymphomas. Okay, and that was just interesting that usually within 4 to 6 months, these mice were overwhelmed by these lymphomas and unfortunately died from that disease. Of course, mice with normal copies of P53 lived their normal expected life expectancy. But interestingly, he found that heterozygous P53 mice had a very different kind of tumor profile. Excuse me. So what he noticed is that P53 heterozygous mice, or mice that are missing one copy of this tumor suppressor gene in every cell of their body, developed a different spectrum of tumors, and actually a large number of sarcomas. About 40% of these mice developing osteosarcoma, as well as soft tissue sarcomas was another large percentage. But about 40% were osteo, with this, notice that at the time, they also had significant genetic similarities to the human disease. So what are the advantages of the genetic engineered mouse models? Well, for one thing, they provide immunocompetent model systems, so you can study some of the genetic effects, the biology, and potentially even therapeutic interventions. And for us as well, they enable a rare disease become common. And that's basically what you can do, is you can actually establish or develop your own cancer center using these particular approaches or genetic engineered mouse models. So as everyone knows, in this particular form, osteosarcomas, there's about 800 to 1,000 new cases of total in the United States of osteosarcoma, with half of them, give or take, being pediatric patients. So obviously that's not a huge number, but what we're able to do is able to kind of genetically, develop genetic engineered mice that in significant numbers. So therefore we have a larger population. So basically, we can have a whole section, or what I call a wing of rats or mice, and each floor, and so we have 9 floors. Each floor can have 7 rooms, and each room can have 5 patients. So just within this one rack of cages, we can have 300 mice with the susceptibility to develop osteosarcoma and potentially even metastatic osteosarcoma. So you can see here the power or the capabilities of the GEMM models. So our design was a little bit different in terms of what Larry did. So Dr. Donauer, he did germline alterations of P53. We decided to be a little more specific because therefore we got able to drive hopefully more osteosarcoma specific development. And so we did specifically P53 alterations in bone precursor cells. So we know that bone cells are derived from mesenchymal stem cells, which go through this pre-osteoblast, osteoblast, and eventually to bone development. So we can actually jump in here and use particular molecular approaches or genetic approaches to alter the P53 status in these specific cells, and that can help drive osteosarcoma. And again, one of the advantages is that these models are immunocompetent or full immune system models that will allow us to understand not only the primary tumor biology, but also the microenvironment as well. So some of the utility of our GEMMs and some of the models and resources we've derived. So we can treat these mice just like our patients, meaning that we can put them through scans, whether it be CT scans, bone scans, PET scans, all this type of X-rays, whatever you want to do. You can do all the same imaging you do on our patients, to the mice as well. Of course, this is showing you a spontaneous. So again, these are spontaneous tumors. There's no cells that are injected into these mice. These are spontaneous tumors that develop in the legs or other long bones typically. And you can see here, this is an osteosarcoma. And so we'll want to be able to sample the pathology, and you can see here representative images that we often see, and they look like the human pathology, where you see these basophilic pleomorphic cells, which are the actual sarcoma cells, and these cells, these tumors are also producing the osteoid, which we see in our human patients. And of course, a subset of our mice will develop metastatic or lung disease, and that's highlighted here as well. So that's the pathology, but of course, from the molecular approach, we want to be able to take the tumors and both the primary tumors as well as resectable lung lesions and look, isolate DNA, RNA, and protein to do our molecular approaches. The other advantages is that we can often isolate blood from these animals, whether it be upfront, at serial blood collections, or at the time of sacrifice, a larger amount of blood. But these bloods or plasma that we can isolate can be used for biomarkers. And I'll show you an example of that actually later on in the talk. We derive cell line models from these tumors, and I'll mention that a little bit later as well. So these are extremely helpful or advantageous in terms of really doing more high throughput studies, especially in vivo studies with some of the combination therapies that we're proposing. And so that leads to kind of the preclinical modeling that we're able to do, not only using the genetic engineering mouse models, but also the cell lines that we've derived from these particular mice. So we've kind of developed, hopefully integrating our mirroring work with obviously some of the patient-derived resources we have, and of course nowadays, using the large amount of human databases that are out there, genetic, genomic and molecular databases, to really develop a pipeline to hopefully identify and functionally study candidate genes and pathways in sarcoma development and progression. And so I'm kind of again showing you examples of some of the genetic engineered mouse model tumors that develop both the primary and metastatic. We can obviously take biopsies of these particular lesions. We can develop cell lines from these particular models. And then, of course, you can do your omic approaches on these particular tumors, whether you want to look at transcriptomic, proteomic, epigenetic, or genomic type of alterations, and you can do an analysis. And so that's kind of the identification portion. But of course, our lab is also interested in, well, what's the functional significance of these alterations? So we can obviously then take whether we do it directly on the GEMM tumors or on the cell lines that we derive, we can put these cell lines back into orthotopically back into the mice, back into wild type C57 black 6. So again, immunocompetent models, study the effects of whether we do genetic alterations on these cells, or we want to knock out a gene or overexpress a gene, or if we're doing small molecule studies, which I'll again highlight here in the next few slides. So what are the effects of whether those molecular or genetic effects or pharmacological interventions have on the primary tumor and of course the metastatic disease? So when we did this, we actually, compiled a large number of our osteosarcoma genetic engineered mouse model tumors. And we looked at what pathways were altered and things like that. And what not so surprising is that we saw a lot of the same pathways altered in our mice and our mouse models that we see obviously in the human disease as well. I'm not going to go through all the specifics or all the details of the molecular analysis. But again, we saw alterations in a lot of the signaling pathways that are involved in proliferation, survival, invasion, and migration, and metastasis. And so some of the receptor tyrosine kinase signaling pathways. And we saw also alterations, enhancement in some of the other pathways, one being the Wnt beta-catenin signaling pathway. And this particular pathway is interesting from the standpoint that you have a membrane receptor that gets activated through binding of its ligand. Eventually what happens is you get beta-catenin, gets kind of released from its complex and translocates to the nucleus, where it leads to transcription factor complex and activation of downstream target genes that are often cancer-promoting genes, such as c-MYC, which is an oncogene, cyclin Ds, which are involved in cell cycle, and other genes as well that are involved in invasion and migration. So these, I'm just highlighting a couple of the pathways because these pathways sparked our interest from a number of different reasons, not only being tumor intrinsic potentially, but also its effect on the tumor microenvironment. So first off, these receptor tyrosine kinase, one being the AXL and MET signaling pathway. So while I highlighted that they are important in the tumor biology themselves, these particular signaling mechanisms also have a role on the tumor microenvironment, immune microenvironment specifically here I'm referring to, including the role of these particular signaling pathways on macrophages, for example, polarization or M1, M2 polarization of macrophages, on myeloid-derived suppressor cells and other cells that are potentially involved in inhibiting particular immune responses to the tumor. And that's kind of summarized here down here as well. So this pathway not only potentially has tumor intrinsic properties, but targeting this pathway could potentially have alterations in the immune microenvironment that could hopefully enhance the activity of that patient's immune profile towards targeting the tumor. And Wnt signaling has more recently gained prominence, not only because of its tumor intrinsic or cell properties, but also what it does on the immune microenvironment. And so it's highlighted here that Wnt signaling or active Wnt beta-catenin signaling can inhibit tumor antigen release, can inhibit or alter antigen presentation, T cell priming, T cell infiltration, and of course, tumor cell elimination from this. So this Wnt activity or Wnt pathway can be very involved in regulating the effects of the tumor immune microenvironment. So with that kind of, both intrinsic and extrinsic factors, we're thinking, well, let's see if we can potentially regulate or alter these particular pathways. So fortunately, there are ways to potentially do this. One of the ways we've looked at is through the use of one drug called Cabozantinib, which inhibits several of those receptor tyrosine kinase pathways that I described before, including the MET and AXL related pathways, and that's kind of schematically shown here, where Cabo is inhibiting some of the VEGFR receptors as well as the MET and AXL signaling pathways. Again, those receptor tyrosine kinases. And those, by blocking those, hopefully you're inhibiting tumor growth, invasion, metastasis. But then what this is not showing is that also at the same time altering the immune microenvironment that can enhance the targeting of the patient's immune system towards the tumor. The other drug we looked at is one that we became involved with over the past several years, and it's called Tegavivint, and this blocks beta-catenin. And so the mechanism of action by which this drug works is that one of the ways beta-catenin translocates into the nucleus is binding a protein called TBL1. And so this TBL1 beta-catenin interaction, again, helps translocate the beta-catenin into the nucleus, allows it to then, the beta-catenin to bind to its transcriptional factor complexes, and then eventually lead to turning on the downstream target genes I referred to before. Well, this particular drug, Tegavivint, blocks or binds that binding pocket between TBL1 and beta-catenin. Therefore, eventually leads to beta-catenin degradation, and then of course you don't get turning on of those genes, those cancer-causing or metastatic promoting genes that are downstream of this particular pathway. So those are kind of the mechanisms of action of these particular drugs. Again, looking at how they're working in the cell. So that I can, go over more background data, but for example, Cabozantinib in the tumor microenvironment, it's been shown that Cabo can induce certain aspects of the tumor cell where it's enhancing class 1 expression, which is involved in allowing T-cell mediated killing, and that was shown in vitro. But then Cabo has actually been used in some murine models as well, one being a colon cancer model, as well as another being a prostate cancer model. And it's been shown that Cabo can increase CD8 or these T cells that are potentially important for killing tumor cells. And that was kind of basically shown in both of these particular models. Now, the data on the Tegavivint is not as published, but it's because the drug hasn't been out there as long. But I think I'm going to show you hopefully some interesting data that we've accumulated over the past year or so, highlighting the effects of targeting the beta-catenin and allowing hopefully increased T-cell infiltration and other immune cells that can hopefully fight the tumor or osteosarcoma cells. So why, again, one of the reasons we're looking at a number of different potential candidate drugs, but these, again, drugs came to the forefront. And again, one of the reasons is because mechanistically, we think they're involved in osteosarcoma, not only intrinsically, but potentially extrinsically. And of course, what their potential translational use in pediatrics. So we know that Cabozantinib, with the Cabo trial that was published back in 2020, there was, a reasonable objective response, and a number of patients, obviously a significant number of patients had prolonged non-progression of their disease. And there's the development of the 2032 trial, bringing in Cabozantinib into upfront therapy with chemotherapy. So it's going to be used. I know at Texas Children's, we were already using Cabozantinib, off trial for some patients with osteosarcoma. And of course, now there's a formal trial looking at Cabo in pediatrics. And with regards to Tegavivint, our lab, and as well as other labs across the country, were critical in terms of developing the preclinical data. This data that I'm showing you down here was from our recent JNCI paper in 2019. But point being is that a lot of preclinical studies from our lab, but others did, showing that Tegavivint was active in a number of different malignancies, including sarcomas. And then my colleagues at Texas Children's work closely with Alterations Therapeutics, which is the company that makes this particular agent, in terms of designing, developing, and eventually implementing the trial, which just opened last October, in terms of Tegavivint for children, adolescents, and young adults with refractory and relapsed disease. So therefore, again, we think mechanistically it makes sense. There's a pediatric relevance that we can easily translate to. And so that made us think about, can we potentially look at these drugs in combination with immune modulatory approaches? So now I'm going to kind of shift, towards some more of the preliminary data that we have that was encouraging, and hopefully that we're going to build on within this proposal. So first off, I just want to show you our Tegavivint data. Again, since this agent's a little bit less known than the Cabozantinib. So we showed in our recent paper that, again, taking metastatic osteosarcoma cells in this LM7 line, which is a lung metastasis line, we injected into the tibia, allowed the tumor to grow up, and then we randomized them either the placebo or the beta-catenin inhibitor, TBL1 beta-catenin inhibitor known as Tegavivint. And we saw the controls grew very well. The Tegavivint was very effective in targeting the disease, both at the primary site, but also at the lung lesions. So we saw a significant reduction in metastatic lesions. And that's using kind of an established cell line. We were also able to obtain lung metastasis from a patient, a relapsed metastatic patient who unfortunately passed away, but we were able to develop the PDX model, did a tail vein injection. And during that tail vein injection, basically we allowed we treated the mice for 4 weeks, either with placebo or Tegavivint, allowed a washout period for about 4 weeks, and then eventually sacrificed the mice. And as you can see here, the control mice had a significant number of metastatic lesions, while the Tegavivint or beta-catenin inhibitor had a significant reduction in the number of metastatic lesions. So again, these are our human models, both the cell line and PDX relevant models. We now want to see, well, can this Tegavivint have an effect on our murine models that we developed again? So these are just showing you a few of the lines that we've tested. We've tested others as well. So these are just individual murine osteosarcoma lines we developed from our genetic engineered mouse model tumors. And so you can see here, we first off wanted to do in vitro testing. So just in tissue culture, just testing the cells on the plate and see how sensitive they were to this Tegavivint. And you can see here that overall these cells are relatively sensitive, relatively low nanomolar concentrations in terms of their effective killing. And not only we wanted to see, well, could it kill the cells, but are we still hitting the target? Okay. And so you can see here the cells that are treated with Tegavivint, we see a loss of the beta-catenin protein. This is actually whole cell lysates, I think. I'll have to go back and check, but loss of the beta-catenin protein within these cells that are treated with Tegavivint, kind of mechanistically showing that the drug is doing its job in terms of decreasing the beta-catenin levels. So then, of course, we wanted to take that in vivo. And so we did a simple, we've tested a number of models now. I'm going to show you one here, but again, Tegavivint had anti-tumor activity when we transplanted the mice. So we took our murine cell lines, our murine osteosarcoma cell lines, put them into the tibia of mice, of C57 black 6 mice. So again, these are immunocompetent mice, allowed a tumor to grow up, become palpable, and then again, randomize them to the vehicle or the beta-catenin inhibitor, and we saw anti-tumor activity here. Then interestingly, we isolated the tumors at the endpoint here, that after 3 weeks of treatment, okay? And what I'm showing you here on the left-hand side, so after we isolated the tumors, we actually did some molecular analysis. So we did what's called transcriptomic analysis or RNA sequencing analysis. And these are just of the whole tumors, okay? And so what you can see here is that when we compared the Tegavivint treated tumors to the control tumors, we saw some interesting kind of patterns or classes of genes that were altered. First off, I wanted to highlight the genes or pathways that were inhibited, which is annotated by the negative score. So these are pathways that are getting downregulated or inhibited in our drug treatment or Tegavivint treatment. So the good news is that we're seeing the Wnt signaling go down, which again, mechanistically, it means that it seems like the drug is hitting its target in vivo, okay? We're also seeing mTOR signaling go down, as well as cell cycles, which is kind of generic, but again, showing that we're inhibiting the tumor cell progression from a molecular profile as well. Not only did we see it, from phenotypically, but again, the mechanism is hopefully there. Interestingly, when we looked at the pathways or genes that are going up, they kind of fell into these pathways that are, again, very immune oriented. So we're seeing these T cell receptor signaling pathways go up, natural killer cell mediated pathways go up. So a lot of these signaling pathways are going, immune regulatory pathways are going up in the beta-catenin inhibitor or Tegavivint inhibited tumors. So with that said, we want to say, well, let's just take a quick look at the tumors. So we did some immunohistochemistry, and we saw that there was evidence that there is some CD8 or T cell lymphocyte infiltration into the tumors with the Tegavivint treated. And we did a quick kind of fundamental FACS analysis, meaning that we took some of the tumors and just sorted the cells and looked for evidence of the CD8 cells and or natural killer cells. And interestingly, in the Tegavivint treated tumors, we saw an increase in CD8 cells as well as natural killer cells, which kind of again corresponded to what we see from a molecular standpoint. Okay, so with this evidence here, as well as again Cabo, which I'll get to in a little bit, I want to take back one last thing we did. Sorry about that. Is that we actually, as I mentioned before, we can utilize these mice and we can isolate blood from these animals to look for potential biomarkers. So we actually did that in a couple of our studies, and one of them being took the Tegavivint treated mice as well as the control treated mice, and took blood and had plasma and sent off plasma from the blood of these animals, of these treated animals, looking for any potential cytokines or chemokines. So these are kind of systemic circulating molecules that could be involved in regulating some of the behavior or responsiveness to the drug. So this is just showing you a couple of our models, but point being is that when we looked at the control mice versus the Tegavivint treated mice, we saw that for example, one of the cytokines or interleukins that went down was IL-4. And IL-4 is known to inhibit anti-tumor T cell activity. So the fact that we're decreasing the IL-4, this again systemically with the Tegavivint is interesting, meaning that maybe now we're getting more T cell activation or allowing more T cell activation with the Tegavivint treated. And one of the chemokines that went up or was increased in the Tegavivint treated cohort was CXCL9. And so again, we're still looking through these panels, we're still doing more analysis, but this one kind of just showed up interestingly, and it's potentially a cytokine that promotes anti-tumor immune response. So again, hopefully something that's showing evidence that Tegavivint's increasing the response of the host response and allowing it potentially to be geared up towards targeting the tumor. And interestingly, there's a report that elevated CXCL9 in melanoma human patients was correlated with a positive response to anti-PD-1 therapy. Okay, so again, systemic analysis of CXCL9 in human melanoma patients, those with higher levels of CXCL9 had a better response to PD-1 therapy than those with lower levels of CXCL9. Okay, so with that said, we decided, well, okay, I keep saying with that said, but one last piece of evidence with regards to beta-catenin, and it's potentially involved in immune inhibition. So finally, is that sarcoma patients typically have a high beta-catenin score, and those are associated with a low T cell infiltration. Whereas if they have a lower beta-catenin score, they could have potentially a higher T cell infiltration. So basically, this particular report, which was just noted a few years ago, looked at a number of different malignancies and basically correlated high beta-catenin score with lower immune infiltration or higher what's called immune exclusion across a number of cancers, with sarcomas being one of them. Now, they didn't specifically, this is not just looking at osteosarcomas, it's obviously a lot of adult sarcomas. But point being, again, the role of beta-catenin signaling in that T cell or in that immune exclusion. And finally, this is literally the final thing. There was a recent study that was published early last year, a single agent Pembro trial, so checkpoint inhibitor. This is, I think, a European and Scandinavian trial, where they only had 12 patients. But what's interestingly is that, so these were Pembro treated in advanced osteosarcoma. What's interesting, they only had one patient that had, of the 12, that had any kind of response. I'm showing you kind of what they showed as in their report, but pre-treatment versus post-Pembro treatment in this particular patient, so patient number 12. So you can see here there's maybe some reduction in activity or tumor activity within this particular patient in response to the Pembro. Well, they actually had some molecular, they did some transcriptomic or molecular analysis of the upfront tumors or the biopsies of these tumors. And what they showed is that across the panel that they did have for tumors, tumors 4, 9, 3, 1, and of course 12, is that there was only tumor 12 that had low Wnt signaling activity in this particular patient, or in this particular cohort. Okay. Now, this particular patient also had other alterations in signaling, but it was interesting to note that they had low Wnt signaling activity, which may have mean that could have led to some of its responsiveness initially to the Pembro or the checkpoint inhibitor. So again, with that said, we wanted to see, well, does inhibition of beta-catenin or the MET axil signaling pathway enhance immune checkpoint in osteosarcoma? So again, using our resources initially, again, this is all preliminary stuff, data, using our murine osteosarcoma models, we're able to, again, inject into the wild type immunocompetent C57 Black6 mice, we're able to do the proper controls. We're able to set up our own little mini clinical trials. So we have all the proper controls. We're treating them with either with single agent Cabo or the Tegavivint, the single agent checkpoint inhibition, and of course the combination. So we're able to do these studies in a relative, I don't say high throughput, but in a pretty, regimented and streamlined manner. And so I'm just showing you for Cabo and Tegavivint. Potentially down the road, we're looking at other targeted therapies, but these two again came to the forefront. So I'm again looking at using some of our models. Top one's looking at Tegavivint, bottom one's looking at Cabozantinib. So you can see here that we have the controls where the tumors again grew, whether it be dextrose, which is the control for the Tegavivint, or the isotype control, which is an antibody for the PD-1, anti-PD-1. So again, those tumors grew very well. The PD-1 had minimal to no, it was not statistically significant, so kind of like our human patients. The checkpoint inhibitor alone does not really have much of an effect on the tumor growth. The Tegavivint had significant anti-tumor activity, and then when we had combined it with the combination of Tegavivint and PD-1, we see there's potential evidence of kind of leveling off or kind of stable disease for a long period of time using these particular combinations. For Cabozantinib, again, we're testing other models, we're investigating obviously the mechanisms down the road, which is part of the proposal. But then for Cabozantinib, we did the similar thing, just presented in a little different way, where we did the antibody control, the solvent, control. Here's the Cabo. We actually are testing other immune checkpoint inhibitors, one being TIGIT and one being PD-L1, which are also immune checkpoint blockade signaling pathways. And of course, we did the combinations, and we also did a combination of the checkpoint inhibitors as well. And what I want to highlight here is that this particular model seems sensitive to Cabo, which again highlights. So I'm taking this and kind of highlighting over here. So the Cabo grew slightly over time. So it started treatment, grew only slightly over time. But you can notice here we did see a significant difference in tumor volumes in terms of with the combination of the Cabo and the TIGIT that we're using. Look at the TIGIT here, but PD-L1 also had it wasn't statistically significant, but the Cabo and the TIGIT was statistically significant in terms of a reduction in tumor volume, even compared to the Cabo. So then our approach, again, we're going to take our syngenetic models. We have ones that are orthotopically more metastatic in nature. We can also do tail vein models to assess specifically more the effects of the treatment as well as combination on the metastatic disease. We're going to do our approaches where we can monitor and do the appropriate randomization. We'll do our comprehensive necropsies and evaluation of the tumors, resecting and primary metastatic lesions when they're available. And we can also do our own little mini toxicity studies. So we want to make sure that are the mice dealing and handling and dealing with these both single agent and combination therapies. We can monitor their weights, their eating, so their eating habits via their weights. We can also monitor, at the time of sacrifice, looking at their organs to make sure there's no significant toxicities noted with these particular single agents, but of course, combination therapies as well. And so ideally over time here, we're going to have, we'll provide some critical biological and preclinical insights into improving immunotherapeutic approaches for osteosarcoma. Again, using our models is hopefully going to provide these insights, and ideally will provide enough rationale such that whether it be Cabo and a checkpoint inhibitor, Tegavivint and a checkpoint inhibitor could potentially lead to some sort of clinical trial over the next several years. And then mechanistically, we hope to, using our FACS analysis and some other molecular approaches, gain some insights into some of the cellular and molecular changes in the landscape of the tumor secondary to these particular treatment regimens. Ideally, hopefully, maybe getting some signatures that are associated with sensitivity, maybe some signatures that are more associated with resistance, and then if they're associated with those type of things, can we then identify new therapeutic vulnerabilities? So our ability to kind of use these preclinical models in a relatively streamlined or high throughput manner will allow us to, again, we can take tumors at different time points of treatment, all right, which again, during human clinical patient trials is not really feasible nowadays, and then do these, pretty relatively comprehensive studies. And so that's obviously the long-term goal of this as well. So again, a lot of this couldn't be done with, without an outstanding team. This was my lab back at Texas Children's at Baylor College of Medicine. I'm fortunate that, several of these members will be moving with me here to Emory over the coming months. And of course, we're looking to hire a lot of new talented researchers as well into the lab over the next couple months. So I think that's it for now. Thank you, Christina and Vicki, and I will stop for sharing.
Great, thank you so much. That was chock full of information, and I mean, interesting about the study that's being funded by the Outsmarting grant, but also I just know all your work on the GEMM models has been really transformative, and I know, and maybe you can even speak to this a little bit more, but I know your models have been shared with other labs.
Right. Correct.
Which
I should have mentioned, I think I mentioned on one of the slides, I didn't highlight it. I didn't mention it, but it was highlighted on one of the slides. And we'd be happy to share these. We've shared these models with a number of different labs across the country. And again, we'd be happy to share whoever else might be interested. So.
Well, I think we actually had Dr. Ligon on from University of Florida last week, and I think they might have benefited from some of your GEMM models for their work, because they're doing some therapy work. So couple questions, and Vicki, feel free to chime in, and also anyone in our audience, feel free to put your questions in the Q&A or even the chat. So just to go back a little bit on some of the basics of the GEMM models. So these models it's spontaneous disease then, right? Because you're knocking out gene, so it's spontaneous disease as opposed to injecting with anything.
Right, correct.
Then once they develop disease, then you create the syngeneic models where you're then
right. So we can then develop, and we develop multiple cell lines or models from the tumors that we can then kind of utilize both from a genetic and pharmacological standpoint.
Yeah. And so specifically you're knocking out one copy of P53, right?
And most of these models, we can knock out both actually too, if we wanted to. But
and
we've done a lot of the heterozygous.
And you touched on this a little bit in one of your slides about how you've noticed then when you're kind of looking, you're doing kind of the RNA-seq and other analysis on these models that you're seeing similar genomic profiles to human osteosarcoma. But I'm wondering how that affects, so if you're knocking out a gene up front, I guess the question is like, do all human osteosarcomas have that one copy of P53 knocked out? And so that's like all the genomic profiles resulting.
So obviously, it's a great question. So obviously P53 is altered in, one way or another in a significant majority of osteosarcoma patients, in terms of within their tumors. And so that's what we're kind of, again, there's no perfect timing. We can potentially improve even the models even more over time, but that we know that P53 is a significant player, or a loss of P53 function is significant in the human disease. And that's what we're just trying to recapitulate to some degree.
Okay, great.
Just a question from the chat from Sabine. Are you hoping this might be a curative treatment or just something to slow down disease progression?
Yeah, that's a great question. I think the hope answer is cure, but again, that's a great question. I don't, we're gonna have to see, as we're investigating more models, and what we're hoping is that if it's only slowing it down, can we identify, again, why is it only slowing down? And through our molecular approaches, can we identify something that will then we can add on or modify that will eventually lead to greater sensitivity and cure, or, of the disease. Right now, the evidence we have, it seems like it's definitely slowing down the tumor progression. Our goal is can we eventually get that to more of a cure?
And another question that I had just about patient 12, you may have explained it and I just didn't understand it, but how come it was just that patient? Like, was there some other genetic disposition they had? Or?
Yeah, so not so unfortunately, it's a great question, Vicki. There wasn't much more information in that particular manuscript about, the molecular or genetic profile of the patients. They had just done what we call, they had some upfront tumor sample from those patients that I showed there, and they did some RNA, transcriptomic and RNA, sequencing analysis on those. And that's what they had seen, is that, when they did the comparison between that one patient that had a response versus the other patients that didn't have response, they saw some alterations in pathways, with the decrease in the Wnt signaling being one of those significant pathways that was noted. Yeah, but I don't know all the other molecular genetic. No, is there something else in those patients' tumors that just wasn't reported? So.
And Dr. Yustein, going back a little bit to kind of maybe ties into Sabine's question, because when I think about, this is also interesting because, normally Cabo's just been thought about as like a targeted therapy, and here you're really thinking about it as kind of priming the immune system a little bit more.
Or both. Yeah.
Yeah,
So
yeah, both.
Right.
And so I kind of think about, like, whenever I think about checkpoint inhibitors, it's kind of like releasing the brakes, right?
Right.
But then there's also like pressing the accelerator. So I'm wondering, like, is there a possibility of the combination of both, or is that like too dangerous? Because then it's just too much going on. But can you kind of release the brakes, but also like press on the accelerator a little bit? Is there possible?
Through what you mean? Through the
Immune system, yeah.
Yeah. So hopefully, again, you're right. It's going to be a balance, and that's why we're going to want to see if there's significant toxicities that are associated. Does the combination really help accelerate the anti-tumor activity? And B, is it accelerating it too much, or you're concerned there's too much else going on, in terms of toxicities? And can we get some evidence or indication if there will be those toxicities using these models as well, right? So I think that's one of the objectives as well.
Another question in the chat from Paul. He asked, will you explore synergy with radiation therapy?
Great question, Paul. We have not thought about that at the time. I know people, it would be potentially interesting, maybe with the radiation causing some, additional neoantigen, production or release of antigens, things like that. We have the capabilities. We've done it before for other tumors. We haven't done it for osteo, but we've done it for other sarcomas that we've modeled. But it's definitely something we can keep in mind. We haven't at this. It's not part of this particular proposal, but it's definitely something we can keep in mind.
Another question in the chat, also from Sabine: if one TKI has failed, might this still be considered as an option in the future?
If one TKI, you mean if the TKI by itself has failed, or?
I think so.
Yeah, maybe. I think maybe there's like two options here. If you've been on a different type of TKI, like let's say regorafenib or something, like,
right,
but this will be an option. And then maybe the other question too is, this has come up in other trials where if you've been on Cabo, let's say by itself, and PAP progression, does it make sense to then try Cabo with another, as a combo therapy with another agent?
Yeah, it's a good question. I don't. I guess it's possible. Yeah, I guess you could think. Yeah, I think each particular RTK could obviously have different. Each patient can respond differently to each RTK. So I guess it's not out of the question that you can try to, a different one depending upon if they failed one. It's not necessarily ruling out this one. So.
And then just again, going back a little bit more to the mouse models, could you clarify a little bit? Because there were a few terms kind of, and I wasn't sure they were kind of similar terms referring to the same thing or if they're different.
Sure.
But when you talk about like the orthotopic injections versus, there's subcutaneous injections, and then you talked about the intratibial versus the tail vein models, are they
right?
Yeah.
So sorry about not clarifying that some more. My apologies. So yeah, when I say orthotopic, that means our intratibial injections. Okay. So we're putting them back into the bone microenvironment and allowing them, and obviously monitoring them there. Tail vein obviously is putting the cells injecting the cells via the vein in the tail. So. And we don't do subcutaneous, actually. We haven't done any subcutaneous models for our sarcoma modeling. So we only either do intratibial or tail vein. So.
And what are the advantages of doing either? Like, why would you do?
So I think with regards to the orthotopic, the intratibial, you're ideally, putting the tumor cells back into a microenvironment that they're more accustomed to, especially if you compare it to a sub-Q injection, And so you can get obviously primary tumor development, and if the tumor cells are highly metastatic, maybe it'll give you some time for metastatic disease, or if you need to do amputation, allow more time to metastasize. Whereas the tail vein is more of a direct metastatic based assay. If you're not interested in the effects on the primary tumor, meaning that the bone, the actual lesion in the bone. So.
Got it. So orthotopic, primary tumor orthotopic.
Yeah, orthotopic means that, so if someone says you're doing orthotopic breast cancer research, it means that they're putting the tumor cells back into the mammary fat pad, for example.
Got it. And then you might have the spontaneous metastasis.
Correct. You still have potential.
Whereas tail vein will give you kind of
direct the metastasis directly. Yeah, you're kind of bypassing the primary, the primary tumor, the primary bone.
Yeah. So interesting combination, but again, like all the backbone that you've kind of developed for all this is really fantastic, just because as you mentioned, you have so many models that you can kind of test things quickly and do the check-ins along the way,
right,
which is difficult to do, right, in some other models. So that's a fantastic setup for further exploration, even beyond the work that this grant is.
Correct, right. Part of the goal is setting up the framework or the pipeline, right? And so being able to do our own little mini clinical trials with whatever approaches we want to do. But obviously, we're very excited about these particular approaches. So.
Yes, very much so. Well, thank you so much. We're just
thank you.
A few minutes here at the end, so we have to wrap up and close up. Thank you so much, Dr. Yustein, for joining us today on Osteobites and making it better for pediatric oncology patients everywhere. More information on this and all Osteobites can be found on YouTube, on our website at mibagents.org, and at your favorite podcast place. And next week, and actually great to have you here, Vicki, to kind of elaborate more on this too. We have our AYA podcast Osteo. We have 4 Osteo Warriors and MIB Agents Junior Advisory Board members, including Vicki, but also Camille, Mia, and Kerrigan. So we have this AYA podcast that drops on the 3rd Thursday of every month. So next week, you can look forward to the 3rd episode of Osteo dropping on August 18th. And then we'll resume with Osteobites on Thursday, the 25th, when we speak with Dr. Kurt Weiss. He is our Outsmarting Osteosarcoma 2021 grant recipient, and he's going to be giving us an update on his funded work exploring aldehyde dehydrogenase and androgen receptor actions to outsmart osteosarcoma metastasis. If you have any ideas for future Osteobites topics, please share them with us. I put an email address in the chat. And thank you again to Dr. Yustein and Vicki for spending an hour with us today, and to our sponsor, the Osteosarcoma Institute. And thank you for joining us today on Osteobites. Check out our YouTube channel or your favorite podcast place for the new episode of Osteo next week. And we hope to see you on the 25th when we talk to Dr. Weiss. And in the meantime, also go to our website to sign up for Outrunning Osteosarcoma. All right, thank you, everyone.